Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Central University's Common Entrance Test MSc Entrance question paper conducted in the year 25th June 2023. This is Biochemistry section that I am explaining. If you are new to my channel, and please do like, share and subscribe. I have also uploaded still previous year question papers of MSc Entrance exam. You can refer to my channel playlist for the same. Next one. In polarized epithelial cells of the PCT of kidney, the difference in apical and basolateral membrane is maintained by. So it is maintained by tight junctions. Then, if a double-stranded DNA has 30% guanine, what will be the percentage of adenine? So as per Chargaff's rule, 30% guanine means it is having 30% cytosine. So total 30 plus 30 will give you 60. 100 minus 60 will give you 40. 40 will be adenine and thymine together. They have asked what is the percentage of adenine. So adenine will be 20%. Answer is option 2. Recognition sequence of restriction endonuclease BAM-H1. Now if you don't know the sequence of BAM-H1, just see for the palindromic sequences. That is, they should read 5 prime same in the 5 prime 3 prime direction in both the strands. So this is 5 prime AAGC TA. This is 5 prime of the complementary strand. It is TAG. So it is not reading the same. Then next one, if you see, it is GGATCC in the first strand. In the second strand also GGATCC in 5 prime 3 prime direction. So 2 is the answer, option 2. Now 3, if you see GTA, it is CTG here. So they are not palindromic. Now fourth one, if you see AGA, TGA, it is in the complementary strand. So that is also not palindromic. So the answer here is option 2. Then next one. Which of the following statement is not correct? The presence of an enzyme has no effect on delta G. Yes, that is correct. At equilibrium, the rate constants of forward and backward reaction are equal. At equilibrium, the rate of forward and reverse reaction are equal, not rate constants. So that is the answer. They have asked which of the statement is not correct. So the answer is option 2. Active site histidyl residues frequently aid catalysis by acting as proton donors and acceptors. Yes, histidine in the active site can act as proton donors and acceptor. So that's correct. Enzyme lower the activation energy for a reaction. That's also correct. Then next one. In hypothetical enzymatic reaction, A giving B catalyzed by enzyme 1, B giving C catalyzed by enzyme 2, the end product C is an allosteric inhibitor of enzyme 1. C will regulate the activity of enzyme 1 by. So the end product of a metabolic pathway, it binds to the enzyme at the start of the pathway. So that is an example of feedback inhibition. Answer is option 2. Then next one. Which one of the following elements bind to proteins and form a critical DNA binding motif? So it is zinc, as in the case of zinc finger motif. Then next one. Kwashiorkor disease is linked with insufficient intake of. So it is due to insufficient intake of proteins. Then next one. Which of the following groups consist of only aromatic compounds? So tyrosine, yes, it is aromatic. Threonine, no. Then next one, if you see phenol is aromatic, tyrosine is aromatic, phenylalanine is aromatic. So answer is option 2. Then next one. Which of the following statement is true? Molecular weight is expressed in Daltons. No, it is molecular mass that is expressed in Daltons. So answer is option 2. Relative molecular weight, relative molecular mass. So they both are not correct. It is not relative molecular weight nor relative molecular mass. It is just molecular mass expressed in Daltons. Now, molarity is a better way of expressing the concentration of solution as compared to gram percent, especially under all conditions for anhydrous molecules. Yes, it is for anhydrous molecules. Molarity is mole based unit, directly expresses the amount of substance present. So for anhydrous molecules and hydrated molecules, molarity would be much better. Then next one, Lineweaver-Burke plot of a viral enzyme in presence of two drugs is shown here. Which of the following statements regarding the action of drug is true? So this is the line for no drugs. This is for drug A and this is for drug B. Now in competitive inhibitor, Vmax remains unchanged and Km decreases. So this value over here, this is your Km. These values which intercept on the y-axis is your Vmax. This x-axis intercept is your Km. So this one, in case of drug A, the Vmax is the same but Km is decreasing. See, Km has come here. It has decreased. This is minus 1 by Km. Okay. So Km has decreased here. It has come to this side. So it is decreasing. Whereas, Km has increased. I am extremely sorry. Km has increased. It is not decrease. It is increase. In case of drug A. But there is no change in the Vmax of drug A. So it is an example of competitive inhibitor. So drug A acts as a competitive inhibitor. Now drug B, here the Vmax is reduced. It is 1 by Vmax. So Vmax is reduced and Km remains unchanged. So it is a case of non-competitive inhibitor. So drug B acts as a non-competitive inhibitor. So that is seen in options A and D. So the answer here is option 2, A and D. Then next one. Given below are two statements. One is assertion, other is reason. Choose the most appropriate answer from the options given below. Now assertion. During starvation, when beta oxidation of fatty acid is predominant in the liver, acetyl-CoA formed enters citric acid cycle. Now acetyl-CoA formed, it doesn't enter citric acid cycle. Instead, it is converted to ketone bodies through ketogenesis, such as ketone bodies such as acetoacetate, beta hydroxyacetate and acetone. It doesn't enter citric acid cycle, and these ketone bodies, they are energy source for brain and heart and other organs during starvation. So first statement, assertion is not correct. So A is not correct. That is seen in option 4. Okay, now acetyl-CoA can combine with oxaloacetate to gain entry to the citric acid cycle. That statement is correct. But it is not any reason for assertion A. Here assertion A itself is not correct. So the answer here is option 4. Now next one. Again assertion, reason. So assertion. Maternal blood oxygen saturation is quite low in the placenta. Yes. Even at this lower saturation, oxygen is readily transferred to fetal blood. That is correct. Fetal hemoglobin binds BPG, 2,3-bisphosphoglyceric acid, less strong, that is BPG, less strongly, thus having higher affinity for oxygen as compared to adult hemoglobin. That is the reason for why even at lower saturation of oxygen is readily transferred to the fetal blood. So the answer here is both A and R are correct. R is the correct explanation of A. Then next one, again assertion, reason. Insulin act on multiple tissues as compared to glucagon. That's correct. It is a protein hormone. Okay, insulin is lipid soluble. No, it is a protein hormone and it is hydrophilic. It is not lipid soluble and easily passes through cell membrane of target cell. That's also not correct. It cannot pass through the cell membrane of target cell. Instead, insulin, it will bind to the receptors on the surface of the target cell and they will trigger a signaling cascade. So A is correct but R is not correct. Answer is option 3. Then next one. Given below are two statements. Again assertion and reason. Assertion. Oxidative phosphorylation. The synthesis of ATP from ADP and inorganic phosphate requires energy. Yes. The energy for oxidative phosphorylation is provided by proton gradient generated during electron transport chain. Exactly. So both A and R are correct and R is the correct explanation of A. That is the answer. Answer is option 1. Then next. Again assertion, reason. Glucose get trapped in extrahepatic tissues. Extrahepatic tissues are your muscle and adipose tissue and does not move out of the cell. Then glucokinase irreversibly phosphorylates glucose to glucose 6-phosphate in extrahepatic tissues. There are no transporters for glucose 6-phosphate and glucose get trapped in extrahepatic tissues. Now A is correct. Glucose get trapped in extrahepatic tissue and does not move out of the cell. That is correct, but R is not correct. Why? Because it is hexokinase and not glucokinase responsible for irreversibly phosphorylating glucose 6-phosphate in extrahepatic tissues, leading to glucose being trapped in extrahepatic tissues. So the answer here is option 3. A is correct but R is not correct. Then again assertion and reason. Salt is added as preservative for storage of meat. Yes, salt creates hypertonic environment and most bacteria cannot grow in hypertonic media. That's correct. So both A and R are correct and R is the correct explanation of A. Answer is 1. Again assertion and reason. Assertion. Restriction endonuclease cleaves the foreign viral DNA inside the bacterial cell, but bacterial DNA is protected from restriction endonuclease attack. Yes, A is correct. Now reason. Bacterial DNA does not contain recognition sequence of restriction endonuclease. That is not correct. They have recognition sequence for restriction endonuclease, but bacteria, they have evolved mechanisms to protect their own DNA from getting digested by their own restriction endonuclease. And one such modification is by methylation of their DNA to prevent cutting. So A is correct but R is not correct. Answer is option. Then next one. Enzymes involved in urea biosynthesis from ammonia are listed below. Arrange these in order of their action. So as you can see from the figure, carbon dioxide plus ammonia by carbamoyl phosphate synthetase getting converted to carbamoyl phosphate, it enters there, then it is getting converted to citrulline by ornithine carbonyl transferase. Then L-aspartate is entering there, then arginosuccinate synthase, it get converted into arginosuccinate, then comes arginosuccinate lyase, then arginase. So the answer here is first will be carbamoyl phosphate. D. So D is there in option 2 and 4. 1 and 3 can be ruled out. Can be omitted also. Then fourth will be D. Carbamoyl phosphate synthetase 1. Then comes ornithine carbamoyl transferase. Carbonyl. Okay. B. Then comes, so answer is option 4. After D it is B. So answer is option 4. Arginosuccinate synthetase E. Then lyase, arginosuccinate lyase C. Then arginase A. So the answer here is D, B, E, C, A. Then next one. Arrange the stages of infection process as they occur. Evasion from the host defense mechanism, entry of the pathogen. That will be the first one. Entry of pathogen. First the pathogen has to enter. So first will be entry of pathogen B. So 2 and 4 can be omitted. So B we will see. Then comes evasion from host defense mechanism. So once the pathogen has entered, it has to evade the host defense mechanism. So A. Then damage to the host cell. Then it has to do its work of damaging. So C. So answer will be option 1. D. Exit from the host. So B, A, C, D is the answer. Then next one. Arrange the following stages of bacterial growth curve chronologically. So as you can see from the figure, it is lag phase, then log phase. That is a exponential growth of bacteria in the log phase. Then comes the stationary phase, and then comes the death phase. So the answer here will be A, C, B, D. A is lag phase, then log phase, then stationary phase, and finally death phase. So the answer is option 4. Then again, arrange the steps in processing and presentation of endogenous antigen by an infected body cell as they occur. So it is endogenous antigen. I already told you in one of the previous question, endogenous antigens are processed and presented by MHC class 1 molecules. So first one will be digestion of antigen into peptide fragments B. Then comes synthesis of MHC1 molecule. Then comes binding of the peptide fragment to MHC1 molecule. Then packaging of antigen MHC1 molecule. Insertion of antigen MHC1 complex into plasma membrane. So the answer here is option 3 B, C, A, D, E. Then next one. Gene expression is controlled at various stages written below. Arrange these stages in chronological order. So first will be epigenetic regulation D. Then comes transcription, then post-transcription, that is C, then translation, that is B, then post-translation, that is E. So the answer here is option 4. D, A, C, B, E. Next one. Proteins involved in DNA replication are written below. Arrange them chronologically in terms of function. So first one will be helicase C. Then comes primase. Then comes DNA polymerase 3. Then comes DNA polymerase 1. And finally it will be ligase. So the answer here is option 2.